for the Deanship of Administration registration. It's really a great honor to have Prof. Jamila Farsi with us today, and without further ado, I'd like to welcome her. Thank you.
Assalamu alaikum wa rahmatullahi wa barakatuh. Welcome to the second session of this morning. It's going to be on bioengineering and stem cell. Our first speaker is Professor Shahid Siddique. Dr. Siddique obtained his PhD for his work on the developmental genetics at Max Planck Institute for Experimental Medicine in Germany and TIFR Bombay. He served as a faculty member at Northwestern University, University of Illinois, and University of Chicago, and contributed to vascular biology and cancer research. He was the head of the department of Toyohashi University of Technology in Japan for 15 years, and a reviewer for the National Institute of Health, and he served as an editorial board for multiple journals on biology and nanotechnology. He contributed for more than 130 publications, obtained 4 international patents, and chaired the innovation conference in the USA and in Japan and India. He's also a founder of McGenome LLC in USA, dedicated to novel drug delivery and discovery. And in 2013, he has joined Al-Qura University College of Dentistry as a professor of human genetics and development. His interests are cancer research and oral biology, including regenerative medicine and stem cells. What's not in this CV, which I Googled it and I found that he is actually won quite few prizes. He was an outstanding scholar, Jawaharlal Nehru Prize for an outstanding scholar, and then for the best student as well in his undergraduate, and he speaks 7 languages. So please welcome with me Professor Siddique.
Assalamu alaikum wa rahmatullahi wa barakatuhu. Nahmadu wa nusalli ala rasulil kareem wa azwajihi zurriyatihi ahli baiti ashabihi ajma'in birahmatika ya arhamar rahimin. Greetings and peace on all of you. I come from the city of Mecca. I am very honored that the organizers, Dr. Muhammad Biari, Dr. Jamal, they have inspired to make this forum a wonderful occasion to be together. This is a unique event for a city that has blessed a fourth of population on this planet for last 1400 years. We are always directed to this beautiful city of Mecca, and we get inspired. So actually in today's talk on stem cells, I will quote a line from Quran. And this verse is أخرج الحية من الميت وأخرج الميت من الحي, which is that your creator is the one who produces life from dead, and he is the one who creates dead from living. So the story of stem cells is exactly like that. From stem cells we have regeneration, we have repair, we have healing, we have business, we have companies, we have health, we have many issues that all of you who are here will think and reflect and inshallah will get advantage of. When you think about innovation, just look at where this Hilton Jeddah is located. Hilton Jeddah is located on the beautiful bay of this lovely sea, the Red Sea, and this Cornish area is filled with beautiful, elegant buildings and the road. In 1983, there was a young man called Cary Mullis. He was driving on California coast, very similar to Cornish, And while driving, he was thinking about his work at the lab. What was his work at the lab? He was in Cetus Corporation, a small company that was trying to build how to make copies of a small DNA. And this Curry Malice, while driving on this beautiful Cornish-like seacoast, thought about, if I have to make a copy of DNA, why don't I just heat it into 2 copies? What a simple idea. Take a copy of a small template of DNA. DNA, as you know, is a double helix. You might have seen as a spiral staircase. Actually, it represents the genome from your mother and from your father. What you have in DNA are the 2 copies, which are representing a father strand and a mother strand. And Curry Malice thought about this, and he said, these 2 DNA molecules are being held together nothing but by hydrogen bond. Hydrogen bond is a very weak bond, but because there are so many of them between adenine and thymine 2 and between cytosine and guanine 3, this holds the DNA double helix together. So Curry Malice, while driving on this Cornish-like highway, he thought, if I just heat the DNA, then these 2 strands will separate. As the DNA will separate, I can add the enzyme to copy it, and then I will have, from 2 copies of a strand, 4 copies, because each strand will be copied from 2 to 4 now. And then if I repeat this process, I can get from 4 to 8. And voila, instead of going home, he came back to the lab after his drive, and he exactly did that. He did the reaction, he heated the reaction, added more DNA polymerase, added more nucleotides, and voila, he was able to make about 4 or 5 cycles or 7 cycles, and then he realized that on the gel, the DNA was much more concentrated than he expected. Subhanallah. So the idea is that very simple creativity ideas come while you're not necessarily at your lab or office, but if you're attached to it, mu'allaq al-qulub, that your heart is associated with it, then you will be able to discover that Edison moment. You will discover that there is something unique. So in 1983 he did that, and this became known as polymerase chain reaction, the PCR. We talked this morning from several speakers on biomarkers which are being recognized by PCR. It's a beautiful technique, very simple technique, and 1993 he was awarded Nobel Prize for that. So the story that I told you has several aspects of innovation and several aspects of creativity, but it also tells you that human mind is capable of thinking tremendous things by just focusing on the issue at hand. So if I have the first slide, please. Can I have the first slide? So today I'm going to talk about bone marrow mesenchymal stem cells, BMMC mediated immunoregulation. What is this long title and what is it about? Basically, let me tell you, the stem cells are very exciting for 2 reasons. Number 1, they can make their own copy into more cells. That's their regenerative capacity. So the good thing about stem cells is that they are reproduced. The second thing is that not only they are reproduced, they can assume, they are so-called pluripotent. Pluripotent means that they can assume in future another new feature that they are coaxed into. For example, a nerve cell from embryonic stem cells can become a neuron, it can become a liver cell, it can become a heart cell, or anything that you think of, a cardiomyocyte and so on and so forth. So the 2 exciting properties are regeneration and the ability to produce new kind of cells. Why I'm talking about this in dental innovation? The idea that as a dentist you are a very privileged class. I must congratulate you because you have the easiest source of getting stem cells. Fortunately, some of the things that have been done are actually coming from dental research, and the work that I'm going to summarize today was basically from the lab of Song Tao Shi, University of California, and he has shown many remarkable things how you can do with stem cells that are coming from bone marrow mesenchyme as well as from dental stem cells. The idea of using stem cells not only healing and regeneration, but also in preservation for future. So if you have a baby niece or a nephew who is 6 or 7 years old and their teeth are breaking, don't throw them away. Don't tell them a fairy story, but preserve them, cryopreserve them for the future, because by that a heart disease, a mental disease, or any other disease can be cured from that broken tooth that a child has. So the beauty of stem cells lies in the fact that they can be used after many years if they are preserved properly in cold storage. So you will be surprised that many, many companies have been formed in America, and well, that's not a surprise, but the bigger surprise is that there are more companies storing these teeth in India than America, and they are making a good fortune by promising, this is only a promise, that these stems can be used in future for autologous regeneration. So let's go through the slides. This is a cartoon of stem cells from the dental pulp. This is the dental pulp area, and these are called DPSCs, or dental pulp stem cells, and they can be very easily harvested, not only from children, but even from adult teeth you can get it. So the remarkable thing is that you can get even from adult teeth. So when you are in your clinician's office, when you are in dentist's office, please remember to get a signature from your patient on a form that says, I allow you to collect this material for future use, so that you cover the IRB, the Research Ethics Committee questions, but then you can preserve that sample for future use, both for research and for innovation. So what can you do with that? Here is an example that if you take that dental pulp cell, you can grow them and you can have cells which can regenerate dentine. So dentine can be regenerated, and this is all published work, so I'm not going to dwell too much. But the idea is that if you start with stem cells, you can create new tissue, and it is possible to do that. Now, the challenge is not just repairing a tooth. It's much bigger and much more important. Many of the fundamental rules that we learn about stem cell biology, they just do not apply only to the teeth, but they give you fundamental information on how stem cells actually work in the body. What are the problems? What are the opportunities? What are the challenges? So here are some of the diseases which are handled by mesenchymal stem cells, and these are graft versus host disease, systemic lupus, inflammatory bowel disease, multiple sclerosis, rheumatoid arthritis. These are some examples of inflammatory diseases which are being studied using mesenchymal stem cells. So I'm going to summarize some work which may sound very technical, but as Einstein said, that a thing can only be made simplified as it is. You cannot make it more simple. So I apologize in advance for some of the slides I'm going to show you, which may look very technical, but I tell you behind them is a lot of work and a lot of future potential. So just share it with me, and I'll be happy to answer and explain that in discussion sessions. So please bear with me these complex slides. So mesenchymal stem cells display profound immunomodulatory properties by inhibiting proliferation and function of several major immune cells, such as dendritic cells, T and B lymphocytes, and natural killer cells. So the idea is again that when you have an infection, when you have an inflammation, there is an interplay of cells which are trying to protect you and the cells which are trying to hurt you. So there are molecules called cytokines which are released, which are both yin and yang, good and bad. And it's the balance of the two that has to be mediated correctly. One of the unique features of many of these immune diseases is, including cancer, that the cells which were supposed to die, they do not die. So cell death is a very important part of life. Kullu nafsun zaiqatul maut. The idea behind that is that everything has a program, and one of the program is death. My colleagues John Sulston in Cambridge, Bob Horowitz, who was working in Cambridge, England, and came to Cambridge, Boston, America, and Sidney Brenner, my teacher, they got the Nobel Prize for discovering this phenomenon of cell death. They found in a small organism called C. elegans that actually there are genes in our genome that we have, humans also have that. This was discovered in a small worm called C. elegans, where there is a gene for programmed cell death, which means that when your cell is born, it already has a genome program that tells the cell when the cell has to die and who is going to kill it. So in the immune system, we have this beautiful play of cells which want to survive and multiply, and the cells which want to be killed or get killed so that a balance is maintained. If you kill all the cells, that's not good. If you do not kill any cell, that is not good. So this is a big challenge, especially in stem cell research, because you have to understand how these two forces interplay. So there are certain pathways. This morning, Dr. Ashley Stevens mentioned, and we heard about NF-kappa B, we heard about TNF-alpha. There are other pathways which are also working in this, but basically these two pathways, the TNF-alpha pathway, NF-kappa B pathway, and interferon gamma pathway are very critical for so-called programmed cell death in these immune systems. So this is a very complex slide. I don't want you to study it too much, but one of the things that is very important is this called fast ligand and fast receptor. These are simple proteins which are on the cell surface. These three proteins come together, so it's a trimer. It's a kind of trinity of three proteins coming together, and when they meet each other, ligand on the cell surface and the receptor on the receiving, when they meet, then they start a chain reaction, and this chain reaction is through many molecules turning on the NF-kappa B or IFN pathway. So the idea is that when you introduce stem cells in a tissue, what are you doing? You're releasing fast ligand, FSL, and that will release molecules to inform the local environment how to treat these stem cells. So let me give you the scenery. Suppose there is a wound. Suppose there is a heart which has just been recovered after heart attack, and you want to give stem cells. When you introduce stem cells to that person, how the tissue of heart is going to decide whether these cells which are coming from outside, should I accept them, not accept them, and who is going to monitor that? Who is organizing? Who is Dr. Muhammad Bayari of that organization called Heart? Who is the man who runs the show? Who is the master? This is a big question in immunology, and these questions have been answered by Dr. Xi's lab. So they find that these are actually fast regulated MCP and apoptotic T cells which are responsible for this thing. So here we have what's called the fast ligand, and they induce apoptosis. Apoptosis means simply programmed cell death. And they kill the cells, and when the T cells die, they release debris. So these are the leftover killed dead cells, and when the dead cell debris are available, they induce the NF-kappa B pathway and TNF signaling. So what you have is, when some cell is received, some cells are killed, and because of their death, a new molecule is released, and that starts a signaling process that teaches you how to receive and how not to receive. So there are two things that I want to simplify here. One is that when you introduce a stem cell in a tissue for healing, it's not that stem cells start doing their job just like that. They have to see what is the microenvironment, where they are being introduced. Is it in the mouth? Is it in the liver? Is it in the lung? And what is the microenvironment of that tissue? And then, when the tissue receives the stem cells, how these cells are going to decide whether the coming cells should be kept or should be killed. So this, Dr. Shi's lab found out that the receiving T cells, called Treg, are the most critical. So if you remove T cells, stem cells will not be able to work at all. They were able to find that the T cells are the ones which are very important. So the T cells are the one which play the most important role. So this is a cell which is dying, and this is engulfed inside a macrophage. So the green cell has been eaten up by the red cell, and that's how you see, and then you get the fragments, and you have the cytokines expressed. So what it does, it induces other cytokines like TGF-beta, and that allows naive cells to release more cytokines called T regulation cells. So here is an example what I've been telling you in molecules. This is the experiment that shows that there was an ulcer on the foot of this person, and this is before the transplantation of the mesenchymal stem cells. 20 days after the mesenchymal stem cells were given, this area becomes inflamed, more red, and pain increases. But in 2 months' time, that it starts healing, because now the cells have been accepted, they have been given their duties, and they are regenerating the tissue, and after 4 months, it looks as if there was no scar at all. So this is a remarkable demonstration of how understanding the basic biology can illustrate the mechanism and the principle of how to heal a wound and how to correct human disease. I can go on forever for many, many examples, but I think this is just illustrating the point. So what is then innovation and how we can apply? So we start with questions from clinics. We use animal model to guide patient and cell type selection. We confirm potential mechanisms in patients, and then we go benchtop and animal study to further explore. So it's like cyclic. We come from the question in the clinic, we do the experiment in the lab, we take a tissue or a small animal, we test the idea, we find the discovery, then we apply on human patients. So it goes on. So we have then another example which is very remarkable, which is the recovery in clavicle bone in the head injury. So here is a model in which a tissue of the head bone has been missing, but with the treatment you can get it repaired by stem cells. So that's another remarkable thing that you could see. So how do you see that a bone is growing? You do these experiments first in mouse, and here is an example that you take bone marrow mesenchymal stem cells, you give infusion, and then you do transplantation, and then you harvest this mice and study the tissue, what is happening. So what is happening is we have a base called hydroxyapatite tricalcium. It's a ceramic powder. This ceramic powder gives a base for stem cells to start working on, and then you can see the red color, which is alizarin staining, which shows that the bone is being formed. So in summary, when you induce these cells, they take a little while because the environment has to study what kind of cells they are, and then when they are accepted, they start releasing molecules that will do the regeneration. So what are the pathways? These pathways are the famous IFN and TNF pathways, and as you say, it's a switch between death and not death. Apoptosis, death; osteogenesis, bone formation. So you have to decide, the cells have to make a choice which pathway they are going to take, whether it's going to be the death pathway or bone formation pathway. So this is how you see the markers which are necessary for, this is TGF-beta pathways, SMAD6, SMAD7 cytokines, which are necessary to understand that. So of all the cytokines, one of the most critical one, turns out, is TNF-alpha. This is the one that controls cell survival very significantly. These are the pathways that I showed you, and here is the example of how this bone recovery happens, and this has been published in Nature Medicine in 2011, Liu et al., and they nicely demonstrate how this principle works very well in that. Even more important thing was that a very common substance that all of us use maybe it's in all our homes. It's called aspirin, sodium salicylate. When you have headache, you say, I need some aspirin. Aspirin is non-steroidal, anti-inflammatory drug, NSAID, and that actually turns on certain pathways, COX-1, COX-2, RUNX, which are actually helpful in regeneration. So just by using aspirin, and this is to what Dr. Sultan spoke this morning, if you're thinking outside the box, you can think about a very simple molecule, simple drug which is sitting next to you, and use it in a very innovative way to create something which is very profound and very important. So here this paper was published in Nature by Liu, and they showed that aspirin actually improves the TNF-alpha and IFN-mediated clavarial repairing. So in summary, in short, I would like to say that actually this mesenchymal stem cell-based tissue engineering requires three components: the donor cells, they can be autologous or heterologous, bioscaffold. I talked to you about hydroxyapatite, calcium, so all the biomaterial person, I see Professor Nadia Badr sitting here, will contribute to this wonderful discovery of how to come with new biomaterial which will provide the right scaffold and provide the microenvironment. And then the recipient T cells, Dr. Syed Shahabuddin, who works on cytokines and all that, immunology. So I would also encourage students here from Al-Qura University to please give attention to Dr. Syed Shahabuddin's lectures, because they may be very important for stem cells. So this trinity, this three play between donor cells, bioscaffold, and recipient cells is really what makes the tissue engineering possible. And understanding these molecules and their working together actually helps us solve many of the riddles. So in summary, I talked to you about orofacial tissue regeneration. I told you that they can be used for immunotherapies. And I told you that the key finding of Dr. Shi's group is that they are governed by T cells. And I thank you very much for your attention. And I enjoyed this wonderful gathering of very diverse people who are here. And I'm very happy that Ministry of Health and Ministry of Education has provided a stimulus for innovation, for growth, for incubation of ideas. And I'm very happy to move from Chicago to the warm weather and the blessed city of Makkah. I cannot thank Alhamdulillah Rabbil Alameen.
Thank you very much, Prof. Shahid. Siddique. Well, he told us that we have the easiest, the least expensive, and non-controversial access for stem cell through the pulp. Now, we move to our next speaker, who is Dr. Paul Anderson. Dr. Paul Anderson is an associate professor at the Dental Physical Science Unit, Institute of Dentistry in London. He received a doctorate from Institute of Dentistry, Queen Mary University of London, and his PhD work considered both the chemical and the physical parameters of enamel and hydroxyapatite dissolution. That result in the formation of surface layer peculiar to these two interesting systems. Dr. Anderson recent published work on the salivary protein staterine demonstrate that it and related peptide can inhibit the dissolution site on hydroxyapatite, and that's going to be his talk tomorrow. Thereby, this will reduce the rate of dissolution for up to 50%. A patent has been filed on the use of this peptide for use in artificial saliva and other dental product as an anti-caries agent for xerostomia patients. Dr. Anderson's other key interest is the role of metal ions, particularly zinc and copper, in reducing kinetics of enamel demineralization. And the title of his talk is The Effect of Metal Ions on Reducing Enamel Dissolution. Please welcome with me Dr. Anderson.
Thank you very much. May I have the first slide, please? Can I say, first of all, thank you to the organizers for inviting me to this Dental Innovation Forum. And also to particularly Dr. Thabit, who picked me up from the airport at a very early hour yesterday morning. Okay, so the title of my talk is The Effect of Metal Ions, and why does this is important in dental innovation? Well, it's not so much the project and title area, but more the interaction with GlaxoSmithKline. I'm an academic by training. I've always worked in a university, but more recently I got involved in collaborations with companies, particularly Glaxo, and I think, as was alluded to this morning, the idea is that by working with different types of people, you actually think differently, and that is that interaction with a company that's involved in selling toothpaste and oral healthcare products that has developed these kind of studies, which are much more patient-based than perhaps the more basic science that I was associated with before. So, in a sense, what I'm talking about here is metal ions. Tomorrow I'm going to talk about proteins. So today is what I call inorganic molecular dentistry. Tomorrow I'll be talking about organic molecular dentistry. Dentistry at the molecular level. So, if I can move on to the next slide. Just quickly about me, and Professor Jamila has just confirmed, has just been through this. I'm a physicist by training. I never thought I would end up working in a dental school, but lo and behold, that is the serendipitous path that has occurred. I was supervised by Professor James Elliott for my PhD, who was particularly interested in hydroxyapatite chemistry, the fundamentals of calcium orthophosphate chemistry. And I'm now a reader in the dental school and interested in hydroxyapatite salivary proteins particularly, and I also run an MSc course in oral biology at the university. Just a bit about Queen Mary. Queen Mary is associated, has a medical school at Barton in London, which is at Whitechapel, and just to put it on the map, I guess there's a pointer here somewhere. Is there a pointer? Oh, is there a pointer here? Anyway, little a shows London on the map there. Little a in the right-hand corner shows that Queen Mary's in the east of London, and the final map on the bottom right shows Queen Mary at point A. And just to put it in perspective, get the pointer to work. The little a is near just where the Olympics were held last year. So let me talk about three metal ions and their effect on demineralization, particularly zinc, strontium, and copper, and relate this to the work on caries, which is a global endemic population. And although fluoride has been effective over the past few years, we have to look at other ions as well, because the challenge clinically is the elimination of the dental caries. So, dental disease is one of the most expensive diseases to treat, more common chronic illness, and still in various parts of the world, caries is still a very significant clinical problem. So we need new innovations to tackle these problems still, and that's really where the interaction with companies like GlaxoSmithKline has been very beneficial. So just a quick review, just for background of the enamel surface. A great proportion of mineral is lost before the surface breaks down, so we have this phenomenon of subsurface demineralization, and the maintenance of the tooth surface layer helps keep its integrity. So it's a key to control the demineral balance prior to breaking down. That is an important strategy to develop. So particularly fluoride has been used to to produce these surface zone effects, but really other systems need investigating. So, enamel is a highly mineralized tissue and consisting of periodic prismatic structures, and it's the inorganic material is a non-stoichiometric defective carbonated apatite. It contains carbonated apatite. It's mechanically very strong, but chemically actually quite vulnerable. So what I want us to think about is, rather than at this bulk level as this picture shows, but the prisms which are shown in blue as it were, and then at the molecular level. What is happening at the molecular level, and how can we can develop systems at the molecular level to protect. So it's a sufficiently porous structure, but protons, acids penetrate into the enamel, releasing calcium phosphate from dissolving enamel crystals. Overall, the structure becomes too weak and the remaining tissue may collapse as a cavity formation. But at the molecular level, it is really the interaction of protons with the hydroxyapatite structure that is the problem. And how can we prevent that? There are trace elements found in enamel, whizzing through, particularly, and there's a range of concentrations given here, but the ones we're interested in here are copper, magnesium, copper, zinc, and strontium, which are quite high concentrations. Sorry. So we know that the carbonate in the hydroxyapatite lattice, which is a diagrammatic at the molecular level here, carbonate destabilizes the hydroxyapatite structure by making the atomic structure less stable, and that increases its dissolution capability. Whereas on the other hand, fluoride, as we know, increases its at molecular level, increases the molecular stability. Also, it is known that atoms like strontium, zinc, and copper also can replace the calcium atoms in the molecule to make that too more stable. So if I start with zinc, zinc is in a number of toothpastes, and I won't say which, at a level of about 1400 parts per million. The sodium fluoride is put there as an anti-caries agent. The zinc is there for plaque control and lowering oral odor. It's well known that zinc has good oral substantivity, i.e. it remains in the mouth for some time, and it also reduces the solubility. So we want to look at what the zinc is also doing in terms of protecting enamel. So does zinc reduce demineralization? How does it interact with hydroxyapatite? And is it actually incorporated into enamel? So the first thing was to use this technique, called scanning microradiography, just to see how and if zinc actually reduces how fast enamel dissolves. And I'll go through this technique in one of the workshops tomorrow. What we can also use, rather than teeth, sometimes we use porous hydroxyapatite blocks to simulate enamel, because it doesn't have the variable composition, porosity, and solubility that does. And we can place these specimens into a small, what we call scanning microradiographic cell, and there's just a picture of a tooth piece in there, and we can change the environment that tooth piece is exposed to, and then see how it dissolves if it's then exposed to acid under a variety of conditions. And this diagram just shows, get the pointer to work, but you can see that an X-ray beam passes through the sample, and through that we can tell how much mineral is lost, and we can change the chemical environment in order to simulate oral conditions as we increase the zinc concentration, particularly from a low concentration of 0.1 parts per million up to about 3,500 parts per million. And we can just keep the pH in that cell at around about pH 4, simulating caries. So we can simulate caries and erosive conditions just with acetic acid at pH 4, pH 2.8 for erosive conditions. And then we can gradually increase the zinc concentration to see what happens to the rate of dissolution as that zinc concentration increases. So we can increase the zinc concentration from 0 parts per million to 5 parts, 10, 15, and so on. And then we can see what happens when we gradually decrease that subsequently. So does the rate of dissolution change immediately as that zinc concentration is changed? So the scanning microradiography system shows us how fast, say, a piece of enamel dissolves, and we can just plot that as a function of time. You can see the mass dropping with time as it's exposed to simulated caries or erosion conditions. We can then inject our metal ion at a particular concentration, and you can see that the rate changes. So we can look at how the rate of dissolution of enamel or hydroxyapatite changes as we change the metal ion concentration within this caries or erosive simulating conditions. This just shows raw data plots. You can see we have lots of data, but as we increase from the top left, which has 0 parts per million to 5 parts per million to 20 parts per million, the gradient gradually decreases, i.e. that the zinc is actually inhibiting the dissolution. So we find that as we increase the zinc concentration, basically the rate of dissolution dissolves. And then just starting with that, say at 20 parts per million, oops, where's it gone? Parts per million, we can see it increases. Oh, my graph has gone. Sorry. Yes. And then if we then subsequently decrease that zinc concentration, you can see in blue the rate gradually comes back up again to where it started. So that means the zinc is having a really temporary effect. It's not being incorporated into the system, it's just remaining there, acting at whatever concentration it's working, say at 0, 5, 10 or 15, and then gradually has less effect as the zinc is removed. So you can see the zinc is not staying there permanently, and that's the key point about this. Likewise, with erosion, we find we get that similar solution just with erosive conditions, pH 2.8. Again, as we increase the zinc concentration, the rate of demineralization is slowed down, but as we increase back or decrease the zinc concentration subsequently, the rate goes back up again. So again, it's not a permanent effect. And just look at one particular example, we actually find there's a log linear relationship between how much zinc we have and how much the rate of demineralization is reduced, so that at very high concentrations of zinc, we get almost 100% decrease in the rate of mineral loss. See, that's fairly linear with time. So, how's zinc actually working? Well, we use a technique called ultra-low energy secondary ion mass spectrometry, and this shows us what is happening at the surface and the surface of the enamel. So you can probe the surface of enamel that's been exposed to zinc with a primary ion beam of, say, oxygen, and you use a mass spectrometer to analyze what comes off from the surface. So again, without going into too much detail, without any zinc present, i.e. the control, as you profile deeper and deeper into the enamel, we find there's very little zinc. But with the zinc test control, you can see as right at the surface, there really is an increased amount of zinc. So it seems the zinc is actually remaining at the surface. And this surface is associated at about 1 unit cell of hydroxyapatite. It isn't going in very far at all. So our model is that zinc is actually just remaining on the surface. So if we do X-ray diffraction studies and ICP optical emission spectroscopy studies, and we try and work out what is actually happening at the surface crystallographically, what we find is that although the calcium ions remain fairly constant, the phosphate ions drop as we increase the zinc concentration. So this means the zinc is reacting with the phosphate to form some compound as that zinc concentration increases. And we also notice that the zinc uptake increases as we increase the zinc concentration. So what's happening is the zinc is reacting with the phosphate. And if we take an X-ray diffraction picture of that, what we see is that a zinc phosphate is being formed. So as we put higher concentrations of zinc into the system, rather than the zinc incorporating with the hydroxyapatite, is actually forming a zinc phosphate phase, probably a material called hopite, which is at the surface. So this is giving us an example of how zinc is working and how we don't need to use such high concentrations of zinc, because the higher the concentration, the more hopite we form. So the X-ray diffraction studies show the zinc phosphate species just sitting on the surface. So zinc has this ability also to reduce the rates of demineralization. I'll just quickly go through some results, similar experiments with strontium. Strontium is also known to be replaced into the hydroxyapatite lattice and also known to have some inhibitory capability. So under Carey's condition, again, as we increase the zinc concentration, the rate of demineralization conditions. But this time, as we decrease, we don't get it going back up the same pattern. So it seems like the strontium is remaining in the structure as the concentration of zinc is decreased. And similarly, with erosive conditions, we also find that although we get the decrease in the demineralization, so the strontium is inhibiting demineralization, as we start to decrease the strontium concentration from 30 to 20 to 10, as shown in blue, down to 5, it no longer goes back up again. It doesn't mirror the decrease. So just for comparison, the zinc, as you increase it, decreases the concentration. As you subsequently decrease the concentration of zinc, the rate increases again. But we don't see this pattern in strontium, so there's clearly two different mechanisms involved. We also see that if fluoride interacts with strontium, we get a synergistic action. So I think if you look at QMAB4 on the left, on the right there, if we just have fluoride alone, we get a decrease. If we have strontium alone, we have a decrease. But combined, so we have a much greater decrease. So the fluoride interacts with the strontium, so we are seeing a synergistic action of strontium. Finally, copper. Copper also has this decrease in the rate. It inhibits the dissolution rate, and then as we decrease the copper concentration subsequently, as shown in blue, that rate then goes back up again, and that's true for both copper and erosive conditions. So, in conclusion, we're suggesting there's two of these molecular effects on enamel. There's a transitory effect which copper and zinc have, where they decrease the rate, but as soon as the metal ion is removed, then that protection disappears. That doesn't happen with strontium. The strontium is a much more permanent effect. For zinc, we know that's now related to the chemistry that's going on at the surface and the formation of these hopite zinc phosphate-like materials. We don't know as yet what happens to strontium or copper. So, in conclusion, metal ions can inhibit enamel dissolution and therefore have karyostatic properties. And higher concentrations of zinc are not really essential, and metal ions can be incorporated into mouthwash. On toothpaste, they already are. And finally, can I thank particularly my supervisor, Professor Jim Elliott, and I have a number of acknowledgements to make down on the right-hand side there, particularly my colleagues Richard Lynch, Gary Burnett, and Charlie Parkinson at Glaxo. Finally, just to remember my other colleague who died earlier this year, Dr. Nathan Cochrane. Thank you very much.
Thank you very much, Dr. Anderson, for the valuable talk. Now we open the floor for discussion. Any question from let's start with the female side. Yes?
Thank you, Prof. Anderson. I'm Dr. Hanadi Margalani, Associate Prof in Operative Dentistry. I would like to just ask about the bioglass, whether it's, I mean, now it's useful for remineralization in comparison with the copper and the strontium and the zinc for surface remineralization of the enamel. And is there any side effect from the copper itself on the human cells?
Sorry, on the side effects of the...
The copper. The copper, is there any effect on the human cells or cytotoxicity?
As far as I'm aware, at high concentrations, yes, but really at these very low concentrations of 5, 10, 15 parts per million, I'm not sure there's evidence there is, but I cannot be completely sure about what impact these low levels of copper might have. I'm not sure if how many of those studies has been done, but it certainly would be before it's introduced further into toothpaste. Certainly zinc and strontium are already added. Copper, not so.
And in comparison to bioglass, what about these materials in remineralization? Do you know of bioglass?
Well, this work is really more on preventative, rather than all the work on bioglass is really, a lot of the work is on delivering calcium and other ions in order to produce remineralization and to, say, inhibit dentinal tubules. There's quite a lot of work on bioglasses to do that. But yes, I mean, other ions can be incorporated into bioglasses and used as carriers. Yes, there's certainly a lot of potential in that carrying ability of bioglasses, particularly, as well as glass ionomers.
There is no recent researches about the incorporation of these materials to the bioglass.
Oh yes, I think there is. Yes, I think there's a lot of potential to look at other molecules, both inorganic and organic, that can be incorporated into, say, bioglasses or glass ionomers as well.
Okay, thank you.
Thank you. Any question from the male side? Yes, go ahead.
Sayyid Shahabuddin from Al-Qura University. My question is directed to Dr. Siddique. So you showed, I have actually two questions. The first question is with regard to the wound of the foot that you showed. Was stem cells used because otherwise that would heal? And the second question with regard to apoptosis, does the mesenchymal stem cells induce apoptosis is a means for its survival?
Thank you. Actually, the foot experiment has autologous bone mesenchymal cells. So the patient's bone marrow cells were removed and used to first grow stem cells in the culture. When these cells were in enough population, they were applied on the foot. So they are autologous cells which are incorporated into the wound, and that promotes healing. These experiments have been done also using heterologous cells, using umbilical cord stem cells from other donors. So although the ideal situation is to have autologous, which is to have your own stem cells. The experiments are in progress where people can use stem cells from other sources, for example, umbilical cord stem cells. So these possibilities exist, but this research is in infancy. There are many questions that have to be addressed, and this is not in a very concrete shape yet. Now, the second question about the signaling of the cytokines is, as I said in my introduction, that this is an interplay between different signaling pathways. And Treg cells turn out to be the most important in the recipient niche to modulate immune response of the incoming stem cells. And so the fast ligand process, the interferon pathway, the TNF alpha, NF kappa B pathway, they are heavily regulated by T cells. So this is the major discovery of Dr. Santao Shi, that the role of T cell regulator of immune response is critical in all this. And the second important discovery is the contribution of aspirin in healing the clavial wound, where this sought to promote those signaling pathways which are conducive for regeneration.
Any more questions? Yes.
Assalamu alaikum. Dr. Muhammad Hassan.
We have a question here.
Dr. Muhammad Hassan, Associate Professor in UQUDENT. I'm asking about the case of cranial reconstruction you showed us early. I'm asking about the age of the patients, and I'm asking, is this technology can be applied to the maxilla or mandible?
Yes. These experiments can, in principle, be extended to other tissues besides that, but this is the very first report that has come. So this is in process. But the remarkable thing is that this can be done. So that actually illustrates that the process is compatible with certain healing process. The challenge, of course, is that different wounds or different physiological targets will have different niche. And so there will not be something that you can just copy and paste one technology to the other and hope that it will work. You'll have to have ab initio, starting from the beginning, all the parameters to work out how to do that. Now, this experiment was originally done in mouse, and the way it was done was there are two kinds of mice, the wild type mice and immunocompromised mice called the nude mice. Nude mice is deficient in immune response, and so that can be tested by giving cells from outside. So from the litter mate, they could get bone mesenchymal stem cells introduced in mice and see which of the cells are critical for that process. And by having T cell receptor knockout mouse, they were able to see that it is the role of T cells. I did not go into technical details, and we will have chance to discuss more in lunch session. But the beauty here is that suggests that it is possible for a clavial wound to be healed using stem cells, although it's a long and slow process. Now, let me warn people about too much enthusiasm about stem cells. On one hand, this technology looks very exciting. There's a lot of money involved, a lot of new companies being formed, but 90% of these ideas are still in testing phase. They are not highly reproducible. There is a lot of caveat. In fact, just recently, there was a paper in Nature by Obokata Haruko from Japan, where she showed that just by giving a stimulus of low pH, pH 5.6, you can induce embryonic hematopoietic cells into stem cells, and these cells can go into any tissue type. So there was certainly a big buzz that you can get stem cells just by changing the pH. Just treat the cells for 30 minutes by lowering the pH, how you can get stem cells. So there is a lot of labs which started immediately working on trying to get stem cells using lowering pH technique. Unfortunately, nobody was able to reproduce her results. So there is a demand that her papers from Nature should be retracted. So I just want to give this word of caution that many techniques in stem cell research, besides being science, is also a piece of art, which means how and who and when is doing that experiment and how this result is being interpreted. So although there is a lot of promise, but as a careful scientist, you should be very, very aware of the fact that these techniques are in infancy, they are developing, and they'll take time to get established. And that's why your innovation and creativity will be needed. Thank you.
In Italy, they regenerate some mandibular defect using the stem cells?
Yes, yes.
And the guys in Spain where they develop trachea?
You are absolutely
So,
right, Dr.
you know,
Farsi.
these are,
Absolutely
it seems
right.
like quite promising area.
Yes, thank you.
Okay, we had a question in here, which we start.
My
Okay?
question is for Dr. Paul. Nowadays, there's a trend for remineralization of enamel using nanohydroxyapatite. First, what do you think, or what's your opinion about this trend? Second, is there any enhancing mechanism for the metal ions for during or enhancing mechanism for the nanohydroxyapatite for remineralization of enamel?
The first question is one of the issues that really is a problem with all remineralization is, do we actually recreate the enamel crystal structure? Does that then create the enamel prisms? So have you actually recreated what amelogenesis had first produced? Or are you just redepositing some form of calcium phosphate in a much more haphazard formation? And that question really still hasn't been answered in terms of what is happening at the molecular level in remineralization. Now, as far as nanoparticles which can be transported into a carious lesion, yes, they can certainly increase the calcium phosphate levels up to such that reprecipitation will occur. But is that reprecipitation, is it regrowing of the enamel crystals themselves, or is there just de novo precipitation of new crystals which will not have the mechanical strength or the properties that the initial that the amelogenesis process has first created? And it's my belief that its remineralization will have to be a much more complex mechanism involving organic molecules, as happens during amelogenesis. But there's a chance to emulate that with modern technologies which can use proteins that can orient crystals, for instance, and so they will actually be able to regrow enamel crystals rather than just deposit calcium phosphates in a really haphazard manner inside the lesion, which is not really repair as more as just putting some calcium phosphate back. The second question, which I have forgotten, sorry, could you repeat the second question, please?
Is there any enhancing mechanism for the metal ions during using of nanohydroxyapatite remineralization? It can enhance the deposition?
I think it's unlikely because these ions are inhibiting the crystal dissolution process mechanism. And crystal dissolution is really quite distinct from crystal growth. So if something inhibits one, it doesn't mean to say it's going to reverse the effect on the other. And I think we've got to be looking at alternative mechanisms rather than for remineralization, whereas for developing to have better protection mechanisms, which in a sense is perhaps a better strategy to adopt, to actually just to ensure or to try and reduce demineralization in the first place, then it's likely that there will be, certainly metal ions will inhibit the dissolution process, but I don't think they will they will promote remineralization. I think we have to look at more organic molecules to look at the reprecipitation of enamel crystallites. Thank you.
Thank you. Any question? Any more questions? Nothing from the male side? Okay. Go ahead.
That we have these great... Assalamu alaikum. Dr. Shahid, Dr. Hawazin Lamfon, Al-Qura University. I'm very proud to know that you have this great research in stem cells in the university, in Al-Qura University. I'm interested in autoimmune diseases. I think we all know that nitric oxide synthase type 2 may play a very essential role in T-cell autoreactive T-cell deletion. But my question is, do you think there is an interference between ionose or nitric oxide synthase type 2 reaction and stem cells, mesenchymal stem cells reaction in the body, so we can enhance these by stem cells instead of nitric oxide synthase type 2?
Thank you. Yes, actually, the results that I showed was of Song Tao Shi's lab. We are having a laboratory at Al-Qura University. We plan to do those stem cell research that I talked, but the results I showed today was from University of California, Dr. Song Tao Shi's work. The interesting thing that you ask is about the interplay of these pathways, where these cells, so the key thing to understand is that every cell has unique set of receptors on it, and it also excretes special molecules. Those are paracrines or autocrines. So some are released from the cells itself or they receive. And this interplay between one type of cell with the other decides which signaling pathway will dominate. So as I said in my talk, the important thing for stem cell research is to understand the niche. Niche means the microenvironment where you are going to grow something. So tomorrow in my talk, I will show how a tooth has been regenerated in a large animal and how that niche has been identified to grow stem cells in proper way. And this interplay between NK cells, between T cells, Treg, CD25 positive, CD3 positive, all of this is very important with respect to the microenvironment where you are doing this work. So this is the most important thing to understand, that same protocol cannot be used in all situations. You have to develop and understand the basic biology before applying the stem cells for a specific problem. Thank you.
Okay, we'll take one more question.
السلام عليكم. محمد إبراهيم, Al-Qura University, Mecca Techno Valley, and the IPMO في Al-Qura. My really comment is that I do hope that the speakers start to emphasize the importance of synergies between different disciplines. I think this is really the new frontiers, that if we can break down the academic barriers between disciplines, which has, I think, killed innovation, we can certainly start to move into new discoveries, and also will give our young Saudis the opportunity to look into these synergies. I do hope they comment on that. Thank you.
Well, I think I'm very proud and privileged to come from Al-Qura University that has taken a very positive, under the leadership of Dr. Nabil Kaushak, our Dean, Dr. Biari, who has encouraged to make a research lab, Dr. Bajamal, who is supervising the development of all these resources. So we are breaking the barriers. The idea is to bring people from different disciplines. I'm trained as a geneticist and molecular biologist. I'm talking to oral biologists, dentists, biophysics persons, IT persons. So you're absolutely right. For the young Saudis, ladies and gentlemen, I encourage to talk to each other, especially in forum like these, and exchange your ideas. Come and do not hesitate. As you might have heard in this morning's talks, that a very simple idea can have a profound meaning if you have perseverance, if you follow it up, if you have passion for following it up. There are resources in the kingdom which are available to you, and you can very easily exploit these if you have that passion to go ahead. So I must say what Dr. Ibrahim is saying, that at Al-Qura University we have an excellent resource of transfer of science and technology into business entrepreneurship. We have Makkah Techno Valley Corporation. these are very encouraging signs. When people used to think about Makkah, they only thought about a Qibla and to pray towards it. But maybe it will also become a center of innovation and technology in due course. So I'm very optimistic, and thank you, Dr. Ibrahim, to say that we should break the discipline barrier and bring people together. Thank you.
Okay, on behalf of the organizing committee for this meeting, we would like to thank both our distinguished speakers, Professor Siddique and Dr. Anderson. Actually, they are not only creative in their scientific area, but Prof. Siddique is a photographer, and Dr. Anderson is a musician, so that's another creative area for both of them. Okay, have a good time.
I thank the chairman for this excellent session, and she deserves a special help because there are not so many female chairmen that we see, so mashallah, thank you.